Hello, everyone. Good morning. Today is Tuesday, March 9, 2021, and it is 9 a.m. Eastern Standard Time. And this is the start of our program for virtual online ART training. Today, we are going to cover Module 2, Advanced Andrology Technique. My name is Ashok Agarwal. I am the director of the Andrology Center. I welcome you all to this program. We are very, very pleased to have you join us. And let me begin by my introduction. Okay, so I'm going to be your host for the next 2.5 hours. So let us begin. This is a brief biography about myself, and without taking too much time, I'm in charge of the Andrology Center here at the Cleveland Clinic and have been there for many years. I'm also the director of the American Center for Reproductive Medicine. I staff in the Department of Urology. We are very proud as a group. Our program is recognized as #1 in terms of the number of publications, the quality of publication, and the reach of our publications in the field of male infertility, andrology, and human-assisted reproduction. We have a very large number of medical textbooks that have been published, and we are very proud that very large number of clinicians, researchers, students have received training in our program from more than 100 countries over the last several years. I want to declare that the organizers and speakers of this program have no conflict of interest. We have not contacted any companies or outside entities for sponsorship. The program that we offer is entirely made possible by support from our center. Webcasting is supported by David Reichling and David Wolfley from the Information Technology Division of the Cleveland Clinic. Thank you to both of them. Further, the course faculty have donated their time for this online training. The program coordinators have volunteered their time for this training course. The names, images, and description of certain products or instruments shown or discussed during our presentations are being used routinely in our Andrology Center. These items are not endorsed by the speakers. Now, let's talk about what we are going to be doing today. So today we are going to be teaching and training you in advanced andrology techniques. And the techniques that we are going to cover, we are going to start by discussing what is advanced sperm function test. Then we are going to go to the most important test, which is currently being employed, is called sperm DNA fragmentation test. We'll also cover test for oxidative stress as oxidation reduction potential test. Then we are going to talk about the lab developed test for sperm DNA fragmentation, and then cover the clinical relevance of sperm DNA fragmentation in human assisted reproduction. And lastly, there will be a talk by our clinician on the clinical interpretation of everything that we are going to be covering here. So these are, again, the topics from starting in a clockwise manner: a laboratory evaluation, clinical relevance, evaluation of SDF, and lastly, clinical interpretation of laboratory tests, laboratory results that you will receive, and how to make sense of it. And this is the program in a on the left side, and our speakers for the day here. Our speakers are Professor Ralph Henkel, Dr. Sejal Gupta, Dr. Renata, Dr. Rakesh Sharma, and Dr. Neil Parekh. So today we are going to be talking about the advanced andrology techniques. On the left side of the panel, you see the basic test, which can be covered under the semen analysis and many of the ancillary tests that you heard last in the month of January during module 1. So these are basic tests. And then we are talking about the advanced andrology test. And there are a variety of tests. This panel has just a small number of tests, which we have illustrated here. And many of them, or some of them, will be highlighted in today's talk. So why we chose the test for oxidative stress? And the reason is that there is such a large amount of information on the role of oxidative stress in male reproduction and male fertility. Right from the very beginning, from 1940 to current, there are thousands, almost close to 1,200 publications, which is probably the largest number of publications in one subject matter. So it is a very important area, and we felt that it is important for us to cover that. And also we, on the right side, you can see we have a great deal of expertise in this particular area, and we feel that we can do a very good job in talking about and explaining the practical side of these tests for male infertility. And then the sperm DNA fragmentation, another important test. So if you look at the left side, again, this is a very hot area with very large number of publications, both original publication reviews and meta-analysis. And this data is only until 2019, and certainly the number have increased since then. And on the right side, again, we have at the Cleveland Clinic a great deal of expertise in this area, not only from a research point of view, but also from a clinical point of view. We have developed techniques, we have standardized those techniques for sperm DNA fragmentation testing, and we felt that we can share these things with a great deal of expertise and confidence. Now we want to move to our next talk, and the next speaker is Dr. Renata Finelli. Renata is currently, she joined about 3 months ago as a researcher in our center at Cleveland Clinic. She has a doctorate in biotechnology of reproduction from University of Rome in Italy, and she has received several scholarships and has published a large number of articles and papers in the field of male infertility and human reproduction. She is going to, again, just like Dr. Gupta, who put together the lab results of oxidative stress into clinical context, Renata is going in her presentation talk about the clinical importance of sperm DNA fragmentation, not only just the tunnel, but all different tests in human reproduction or assisted reproduction. So Renata, please go ahead.
Thank you very much, Dr. Agarwal, for your introduction. Good morning, good afternoon, good evening, everybody. So today, these are the learning objectives of my talk. Today, I'm going to discuss the sperm DNA fragmentation as a marker of male infertility and its association with natural and assisted reproductive outcomes. Particularly, I will discuss the association of this marker with miscarriage and pregnancy failure, and also I will present you the clinical practice guidelines which have been published by our group for sperm DNA fragmentation testing in male infertility. But let's start saying that sperm DNA fragmentation is not the only type of DNA damage which can occur in sperm. Sperm DNA can occur in mitochondria, in telomere. We can observe microdeletion of the Y chromosome as well as epigenetic abnormalities. However, in this talk, I will focus only on sperm DNA fragmentation, as it is the most widely studied types of DNA damage in sperm. And also, there are several studies investigating the association of this type of damage in sperm and clinical reproductive outcomes. So let's move. This is on the left side, as you can see, a picture of DNA. sperm DNA fragmentation can occur at just one or both strands of DNA, resulting in single and double strand breaks. Here in this picture, I illustrate three different mechanisms which can result in sperm DNA fragmentation. For example, defective chromatin condensation, abortive apoptotic process, as well as the oxidative stress. All these three processes can lead to fragmentation at single or double strand breaks. This is particularly relevant in clinics because sperm DNA can be repaired until the third week of spermatogenesis, while there is no repair mechanism during the transit in the epididymis or post-ejaculation. Here on the right side, I report a table from an in vitro studies. In this study, as you can see, there are increased rate of sperm DNA fragmentation, and in the table it is reported its association with reproductive outcomes such as penetration rate, fertilization rate, or blastocyst development. As you can see, increased rates of sperm DNA fragmentation does not affect penetration rate or fertilization rate, while blastocyst development is significantly affected when sperm DNA fragmentation is higher than 88%. This is explained by the fact that the oocyte has limited ability to repair sperm DNA fragmentation, and the damage can be repaired only when it is less than 8%, as it is shown in this in vitro study. We should also consider that in the last years, the oocyte quality is declining worldwide, and this results in a declining capability of the oocyte to repair the sperm DNA damage. In general, high levels of sperm DNA fragmentation have been observed to be higher in infertile population, but however, a weak correlation has been reported between this parameter and the conventional semen parameters, which can be analyzed through the standard semen analysis, such as concentration, morphology, or motility. Here in this table, I reported the results from just 3 studies, but there is a huge literature showing this poor correlation. And this is explained by the fact that SDF is an advanced molecular marker of sperm quality. It is a molecular marker, and it is important. It is independent of conventional semen parameter. This is why these parameters should be analyzed along with the standard semen analysis. But let's move on natural pregnancy, the impact of sperm DNA fragmentation on natural pregnancy. In this slide, I reported the results of a meta-analysis published in 2011 by Zini. In this meta-analysis, the 3 studies were included for more than 600 couples, as you can see in the table here on the right side. And this meta-analysis showed that high levels of sperm DNA fragmentation were associated with the failure to achieve natural pregnancy. However, we should not forget that this meta-analysis includes only 3 studies, a very small number, and also the confidence interval per single study is very large, suggesting a poor precision of the sperm DNA fragmentation in predicting the natural pregnancy. Here, in this slide, I report the results from another meta-analysis published in 2019, very recently, and including a large number of studies, 13, for more than 500 men with a history of recurrent pregnancy loss. Here on the right side, you can see the forest plot from this meta-analysis. And this forest plot shows that couples in all the studies included, couples with a history of idiopathic recurrent pregnancy loss have higher levels of SDF than fertile couples. And here I'm not reporting the second forest plot, but the similar results were also shared by another meta-analysis published in the same year. Moving to the intrauterine insemination, a meta-analysis published 2 years ago, including almost 3,000 IUI cycles, reported an association of high and negative association of high levels of SDF and the lower pregnancy and delivery rate when IUI was performed. Here on the right side, you can see the forest plot illustrating the association of SDF and pregnancy rate, while on the right side, the forest plot illustrating the association of SDF and delivery rate. When IVF, in vitro fertilization, was performed, 4 different meta-analyses reported an association of high levels of SDF and lower pregnancy rates. In this table, I summarized the main findings from these 4 meta-analyses, such as also I reported the number of studies included, the number of cycles analyzed, and the main findings. And all 4 agree in reporting this negative association with the pregnancy rate when in vitro fertilization was performed. Also, when IVF was performed, a miscarriage rate was reported to be associated with high levels of SDF by 3 different meta-analyses. Here at the bottom, you can find the references, while on the left side, I reported just as an illustration the forest plot by Zhao et al. Published in 2014. Regarding the live birth rate was reported to be significantly lower in men with high levels of sperm DNA fragmentation after IVF. When ICSI was performed, miscarriage rate was also reported to be significantly increased when sperm DNA fragmentation was equal or higher than 30%. Also, the neutral comet assay was suggested to be a good predictive technique for recurrent miscarriage. As you can see here in the ROC analysis, it showed high sensitivity and high specificity for predicting this outcome, miscarriage rate. Further, live birth rate, the association of live birth rate and SDF was analyzed in a meta-analysis published in 2015 and including 5 studies for more than 400 cycles. And this meta-analysis reported that men with low sperm DNA fragmentation had significantly higher live birth rate. However, we should not forget that all these studies that I've studied so far are not free of limitations. They are heterogeneous in terms of the studies included. They consider different threshold levels for defining high and low sperm DNA fragmentation. Sample sizes are always relatively small. An important consideration is that sperm DNA fragmentation is always measured in semen before the initiation of cycles. This means that the rate of SDF, which is measured, may not really reflect the status of sperm using for insemination. And also, in all these studies, bias and confounding factors are never taken into consideration when the results are analyzed. So let's summarize the current findings so far, described so far. In general, high levels of SDF have been associated with decreased pregnancy rate after in vitro fertilization and with increased miscarriage rate after. This can be explained by the fact that in ICSI, the natural fertilization process is totally bypassed by the embryologist. Also, it is possible that high quality oocytes may counterbalance, may offset the negative impact of sperm DNA fragmentation on pregnancy. Also, we should remember that in ICSI, gametes are cultured for less time than in IVF. This means that they are exposed to oxidative stress and laboratory-induced damage for less time. And another consideration is that DNA damage in sperm may affect genes which are responsible for embryo development and implantation, resulting in an increased miscarriage rate. So now I want to present you the clinical practice guidelines that have been published in last year in 2020 by our group. These guidelines were the results of a collaborative effort from 24 international experts in male infertility, and they were published based on the evidence already published in the literature for SDF testing. In these guidelines, we identified 5 different conditions, 5 different clinical scenarios which may really benefit from SDF testing. And these include recurrent miscarriage, lifestyle risk factors, varicocele, recurrent IUI or IVF failure, as well as unexplained and idiopathic infertility. For all these conditions, different type of treatment can be considered if SDF is significantly higher. And these type of treatments are summarized here in the picture. They include recurrent ejaculation, antioxidant therapy, lifestyle modifications, ICSI and the assisted reproductive techniques, or varicocelectomy in case of varicocele. So these are my take-home message. Don't forget that sperm DNA fragmentation is a functional marker of semen quality, of sperm chromatin quality. It cannot be analyzed by conventional semen analysis. High levels of fragmentation are associated with a longer time to achieve a natural pregnancy, as well as higher miscarriage rate. Also, high levels were associated with decreased pregnancy rate after IUI and IVF, and with increased miscarriage rate following IVF and ICSI. Finally, we provide clinical practice guidelines for the identification of those conditions which can really benefit from SDF testing. So thank you very much. This is a picture from our group in front of the Andrology Center, and this is a picture from the Cleveland Clinic. Thank you very much for the attention.
Lastly, you can connect with our center by sending a request, searching my name and Cleveland Clinic. And the benefit of connecting is that we are providing daily updates on our research activities. We provide free access to hundreds of research articles published by our center, by our scientists and researchers and our collaborators. You can also read the latest research which have been presented at important national and international meetings, reproductive meetings and urological meetings. Learn about virtual research opportunities that our center provides. Learn about our online ART training courses, and also the opportunities to collaborate with a team of ACRM researchers. So please consider joining on our LinkedIn.